This week you're going to be doing the acid-fast stain, and then later you're going to be doing the endospore stain. They both have very similar, not procedures, but supplies that you're going to use. So with the acid-fast stain, last time we did a gram stain. So remember that most bacteria are either gram-positive or gram-negative. There's bacteria that cannot be stained using the gram stain because their walls are impermeable to most stains. And these particular organisms are called acid-fast bacteria. Acid-fast bacteria fall into a couple of genus. The most important one, or the most common that you might be familiar with, is the genus Mycobacterium. In the genus Mycobacterium, there's Mycobacterium tuberculosis that obviously causes tuberculosis, and Mycobacterium leprae, which causes leprosy. These two bacteria have walls, so they have their normal peptidoglycan cell wall like you would see with any other bacteria, but then outside of their peptidoglycan wall, they have a waxy coat, and that waxy coat is called mycolic acid. And that mycolic acid helps protect those bacteria so that they can invade the body get through the defense mechanisms. The other thing about the fact that that mycolic acid is there is if you use something like the gram stain where you put on the crystal violet and then rinse it and then use the ethyl alcohol to decolorize, that purple stain will never penetrate through the waxy mycolic acid. Therefore, it would never gram stain purple even if it is an acid fast, even if it is gram positive. And these are gram positive even though they won't stain that way. So one of the things that I want you to think about as we go through these staining procedures is keeping them independent of one another. With the gram stain, remember that the purple meant positive and pink or red meant negative. Well, with the acid-fast stain, red is going to be acid-fast positive, blue will be negative. And then when you go to do the endospore stain, green means endospore, red is just like the secondary color that you're going to get of the wall. So red doesn't always mean negative. It depends on which procedure you're using. So make sure that you are independent when you're thinking of what stain am I using, what do the results mean. So with that being said, let's talk a little bit about the lab that we're doing. So the supplies that we're going to need are going to be our slides, our inoculating loop like with all the other ones. Now I don't have a Bunsen burner out here because I'm not going to demonstrate how to fix your smear. You already did that in your simple stains, and then you also did that in the gram stain. So remember what I said the first video, is that unless the procedure tells you not to, you're going to be fixing the smear. And it will always say at the beginning of the procedure, make and fix a slide of this particular bacteria. That means you put the bacteria on there using your sterile technique with your loop and your Bunsen burner, allow it to dry, run it through the flame. That's the fixing procedure. So you would have done that already before you start any of this. One of the things that I want to point out about the lab that we're doing this week is that I am making some changes to what you're going to see in the lab manual. So we're going to do it out of the lab manual, but this video, I want you to note the changes because you're going to do it based on the changes that I tell you, but you're still going to be using your lab manual as a resource. So one of the things that I also want to point out is the type of bacteria that we're going to have. The Mycobacterium, because of that waxy coat, the bacteria really stick to each other. So when you go to make a smear, you may have a problem kind of breaking the cells up instead of getting a big chunk of them, and we've had some issues with other bacteria with that happening with. Now, depending on how they grow, Mycobacterium is different from a lot of bacteria in the fact that it grows really slow. Some of them can take several weeks to grow. This one we're hoping will grow up in about 4 days. So if they grow up properly, they're going to be in a normal broth tube like we're used to seeing. So you would just do your flicking, pull the bacteria out using your aseptic technique, and put it on your slide. Wow. If not, if they don't grow up, we have a backup plan. Then this is the way we've done it in the past. Is you would actually take the bacteria off of this slant here. You'll notice some differences. The media is a blue color. It's a special media just for Mycobacterium. You'll also notice the bacteria is kind of yellow in color, and you can actually see the chunkiness of it. So if we have to use these, Just be really careful that when you take the bacteria off the top, you don't gouge the media. I wanted to demonstrate that people have used this in the past and did gouge the media. You want to get only bacteria, because otherwise you're going to be staining the media and that's not going to look like bacteria. Hopefully it works out to where we're going to use these, but if not, we will use these. Because it's taking from a solid, you'll have to do that technique of putting the water in first, adding the bacteria, and then spread it around so that you get a single layer of bacteria. Once again, because it's kind of chunky, that's going to be a little bit more difficult than what we've seen in the past. Now the changes that I'm going to make, what doesn't change is that you prepare and fix your slide. So whether it's from here or from here, you do the same procedure for the fixing. The changes that I'm making are going to be in the actual procedure of the staining. So in the staining, in our lab manual, it just says to flood the smear with the carbon fusion and allow it to sit for 5 minutes. I'm going to use a different procedure, and we're going to use heat to try to get the stain to actually penetrate the slide, to penetrate the bacteria. Because of that waxy coat, it's really hard for the stain to actually penetrate the wall of it. So that's going to be the first change that you're going to see. So when you go to do that, you're going to have a boiling water bath in front of you. Now it doesn't need to be any kind of a rapid boil or anything like that. So once it starts to boil, we can kind of turn it down. So one of the things you want to make note of is it does take a while for water to boil. So before you ever get started on the labs of making anything, get your water bath going, because it takes a while to get started. Once it's going, like I said, you can turn it down, because basically if there's steam coming off of it, that means that it's hot enough and it's going to help the heat penetrate, help the stain penetrate due to the heat. So I have my bacteria already prepared, already fixed on the slide. Now when doing this, be careful because it is hot and you don't want to burn yourself. So what you're going to do is you're actually going to place the slide on top of the actual boiling water and have the steam and warming the slide. Now more than one person can do it at a time. The key is to just be very careful and not to burn yourself or knock the slides into the water. Our goal is not to make the water change color. Our goal is for the slide itself, for the stain to penetrate. So what you're going to do, you put the slide up here after you fix the smear, but before you put the stain on. So now you're going to add the stain, and you're just going to flood it like you've done with previous labs. So you're just going to put it on there. What you want to make sure doesn't happen is that it dries out. So you're going to put it on there, and it says to let it sit for 5 minutes, and you are going to let it sit for 5 minutes. While this is steaming for 5 minutes, you want to keep an eye on it, because if it dries out, it's not going to stain properly. So as it's going along steaming for 5 minutes, keep an eye. If it looks like it's drying, add some more stain. Okay? Try not to let it run over too much. Like I said, just enough to cover it, but not the water's turning color. We have to clean the beakers, and it's a little difficult when they have too much stain in them. So that's the first change we're making. We're going to actually put the slide on the boiling water bath to steam it so that it allows the carbon fusion to penetrate the wall. So that being said, our first stain in the acid fast is the carbon fusion. So now you're noticing we're using a red stain as our primary stain as opposed to the purple one of the gram stain. So now we'll pretend that our 5 minutes has gone by, and now you want to take the slides off, of course, without burning yourselves. And you can do that by just grabbing the edges and putting them to the side. Okay? Now you're going to take it over to your staining tray. One of the things that we don't want to do is break the slide. And you're probably aware that if you put cold water on hot glass, that it's going to break the glass. So we're going to have to let that cool for a minute before we can start rinsing it. While that's cooling, let's go ahead and just kind of move this guy out of the way and talk about the next step. So that's the carbon fusion for 5 minutes on the steaming. Now this is going to get rinsed off as soon as it's cool enough. So if it's cool to the touch, it's probably cool enough. So I'm going to go ahead and take my water, and I'm going to rinse it just as we did with the gram stain procedure. Okay, just as the same as with the gram stain procedure, the next step Well, actually the next step in the gram stain was the mordant, but we're not going to do one of those here. The next step is going to be the decolorizing. If you recall from the gram stain, the decolorizer was either alcohol or acetone alcohol. In this, it's going to be acid alcohol. The reason that we use acid alcohol is because if a cell is acid fast positive, it won't decolorize even in the presence of acid. Okay, so we're going to use acid alcohol for our decolorization. And so we're going to do just like we did with the gram stain. We're going to just drip on there until the stain runs almost clear. Not clear, but where it's a very light peak. Okay? Just as we did before, we want to stop that process right away. So we're going to rinse it immediately. And once again, we're not drying between any steps. Now we're going to use the secondary stain, which in this case is methylene blue, which you used in the first lab. Your simple smears. Your lab manual says to leave that on for 1 minute. You can go 1, but even up to 2 minutes with your counter stain. So after that's set for its 2 minutes, we're going to rinse it off. And now that we're done rinsing, we can grab a paper towel, which I seem to not have, so pretend I had a paper towel. You would blot the back like we did before, and gently blot the front, but don't wipe your smear off. You don't want to waste your last 10 minutes of work. So that's the acid fast stain. The next stain that you're going to learn is going to be the endospore stain.